three essential functions of the cell. The cytoskeleton is embedded in the protoplasmic matrix. Contraction and expansion of the various organelles of the cell, and in some cases the pseudopodia also.
Thank you for watching this video.
Hello students, welcome to btosacademy classes. So we were studying the structures and functions of cells. Now we will look at another important cell organelle, that is the cytoskeleton. The cytoskeleton is an organelle that is not generally visible, but it is very important to maintain three essential functions of the cell. Number one, to maintain shape of the cell, to maintain shape of the cell. Number two, to provide mechanical support, and number three, in order for a cell to divide, it needs a framework. It needs a structure from where a cell can break off or divide into two daughter cells from a single mother cell, or it can separate into two daughter cells from a single parent cell, as we see in plants. So it also helps in cell division. As in humans or any other animals, we cannot see our skeleton, can we? No, because it is embedded under our skin, our adipose tissue, our muscles, our blood vessels, and everything. Similarly, in a cell, the cytoskeleton is embedded in the protoplasmic matrix. However, the cytoskeleton has three major components. The first component of cytoskeleton is the microfilaments. The microfilaments. These microfilaments are made up of a structure known as actin. This actin is a polymer. Actin also has two parts. One is the monomeric part of the actin. It is known as the G-actin or globular actin, and n number of G-actins make up the filamentous actin or F-actin. Now, n number of F-actins make up the microfilaments. So, to recapitulate, microfilaments are made up of actin. Actin is a polymer, it is a protein. Now, these polymers are made up of G-actin or globular actin monomers. N number of G-actins aggregate together to form the filamentous actin or F-actin, and numerous F-actin aggregate to form the microfilaments. These microfilaments helps in maintaining the cellular shape and also helps in contraction and expansion of the various organelles of the cell, and in some cases the pseudopodia also, as we see in the amoeba. In humans, this actin is also associated with another monomer called the myosin. In humans, there is another thing along with this actin is the myosin. Now this actin-myosin complex is present in our muscles, and it helps in muscle contraction. It helps in muscle contraction by incorporating calcium ions in the endoplasmic reticulum of the muscle cells known as the sarcoplasmic reticulum. So this is all we need to know about the microfilaments. Another thing that we have are the intermediate filaments. Now, there are 50 types of intermediate filaments, and they perform various functions that are not very important in our syllabus, but we should know that the intermediate filaments helps in various movements of the protoplasm, helps in the diffusion and osmosis of the protoplasm, and it also provides structural shape and support to the cell. It helps to maintain the shape of the cell, it provides mechanical strength to the cell, and also the intermediate filaments work as a mesh or network in which various cell organelles can attach themselves in the protoplasm. Now coming to the last part of the cytoskeleton, here we have the microtubules. The microtubules. The microtubules are made up of a protein called tubulin. It is a protein. This tubulin has two monomers, alpha and beta. Now, alpha plus beta dimers forms the tubulin polymer or the tubulin proteins. These are the monomers. Tubulin does various important activities. We should remember one thing, that the length of the tubulin protein is not fixed, as the alpha and beta dimers can be added or subtracted according to the need of the cell or according to the function that particular tubulin protein is going to perform. For example, when the tubulin protein helps in the expansion of the pseudopodia in an amoeba, the length of the tubulin protein increases by incorporating more alpha and beta monomers and by rapid dimerization. However, when the pseudopodia is getting resolved back into the cell, the length of the tubulin proteins are getting reduced by subtracting alpha and beta monomers, and hence by de-dimerization. By de-dimerization, that is shortening of the alpha beta dimer chain. Tubulin also helps in the formation of the mitotic spindle, that is helpful in cell division. The spindle fibers attach to the kinetochore region of the chromosome. If this is a chromosome, this is the centromere, and in this region it is called the kinetochore. This region is called as kinetochore. Here the mitotic spindle fibers attached helping the chromosome to move from one pole to another and to align themselves in the cellular equatorial region during the metaphase. Tubulin also acts as a site of attachment of motor proteins. Now, what are motor proteins? Motor proteins are those proteins that can change their structural conformation and can actually help a cellular part or a part of the cell or an organelle to move from one part or another into the cell, or can help the cell itself to move, like WBC cells, the white blood cells. Whenever there is a wound, whenever there is an infection, the WBCs move into that site of wound or site of infection by a process called diapedasis. It is almost like the crawling of a spider. They crawl across the blood capillaries and they reach the site of the wound by the process of diapedasis. Now, these functions are done by the help of microtubules and the tubulin proteins. Also, microtubules originate from the cell in a region known as, they originate from a region in the cell known as microtubular organizing center or region. In this region, in animal cells, the centromeres are found, and also in prokaryotes, where we find the cilia and the flagella as the locomotory organ, they arise from a region called the basal bodies, called the basal bodies. Now, from these basal bodies, the microtubules arrange themselves into cilia or flagella. However, in that case, when a cell has a flagella, the tubulin protein is not there. Instead, another protein called flagellin, called flagellin, called flagellin is found in case of flagella or flagellate animals. Now, this flagellin incorporates two other proteins, myosin, dynein, nexin. These three classes of proteins, mainly dynein and nexin, myosin to some extent but not that important, mainly nexin and dynein, in order for the flagella or the cilia to move about. So, in short, the cytoskeleton not only gives shape to the cell like our skeleton gives the shape to our bodies, they not only provide the mechanical support or site of attachment of different cell organelles, they not only help in cell division, but they also help in cell movement or cellular locomotion. So, this is all we need to know about the cytoskeleton.